Good evening, everyone. Um, Today, thanks for the invitation, Nikki. Today, I'd like to talk about STPs with zero pumps, zero odor, and no color. Yesterday, the organizers asked me to speak about this topic. So today, I'd like to discuss what we at New Algae Nanobiotech in Bangalore are doing in this field. So we are a small company in Bangalore working on interesting nanotechnologies solutions for nature-based remediation of polluted water bodies as well as STPs. So I'm going to walk you through some slides and then we can have a discussion on this topic. So what is the problem today
Can you please put it in presentation mode?
Yeah, sure. I'll try, but I'm not sure I can share it. Are you able to see it now? Is it bigger?
Yes,
Yeah, we
yes, yes.
can.
Okay.
Yes.
So the problem, just to backtrack a little bit with STPs in India, is that you have high energy consumption from aeration and multiple pumps, right? And you have the inability of these plants all over India to meet the discharge norms in terms of BOD, COD, TSS, TN, TP, fecal coliforms, total coliforms, and so forth. There are certain parameters that are regulated by the Pollution Control Board, and it is becoming increasingly challenged for many of the plants to meet them. And this could be for any number of reasons. There could be load fluctuations, input water quality changes, unexpected mechanical failure, power disruptions, lack of real-time adaptability in the plants, and longer than desired shutdowns for maintenance. So these are some of the issues facing STPs. In addition to this, you also have a high volume of sludge that is generated, and this is becoming quite costly in terms of what is required to dispose of. So just to, I guess, sort of tackle all of these issues, problems, let's start from the beginning, right? So why do STPs smell and turn black? The root cause, when one looks at the subject a bit deeper, is that you have uh, big pockets of the STP where dissolved oxygen collapses for a number of reasons. And when this happens, anaerobic bacteria begin to thrive. I mean, this oxygen collapse could be due to power cuts, equipment failure, sludge buildup, poor mixing of the water, and shock loads, right, in the inflow. So in terms of chemistry, what's happening, right? So when you have low dissolved oxygen, the anaerobic bacteria grow and they start interacting with the sulfates in the water and organic matter to produce hydrogen sulfide, right, which creates smell. Some of this hydrogen sulfide also reacts with iron and produces a black precipitate, right, which is what we call black color, right, in the STP. So this is sort of what is happening at a chemistry level. In terms of timeline, what happens is that sometimes when you have a drop in DO, uh, because of various lack of aeration or whatever, right? So there are reasons. Within a few hours, you have hydrogen sulfide forming, odor starting, and within 24 hours, you have black iron sulfide. Right? And the treatment begins to fail. So this is basically an alarm for us, right? So you have treatment failure, you have worker safety hazard, right? Hydrogen sulfide is toxic. You have a corrosive environment where assets are being corroded, and you also have regulatory violations. So to prevent this, many STPs monitor the DO daily. They maintain their blowers and diffusers. They desludge regularly. They have backup power, and immediate action is taken when odor is first noticed. Right. So some of the good STPs do this, but that is few and far between. So what are some of the other things that STPs do to solve this problem? The typical response to this problem is that you have large blowers to increase the aeration, you put more pumps for recirculation, you have high air diffusion rates. There are STPs that also install odor control systems, right? These could be ozone oriented, activated carbon scrubbers, biofilters, and chemical scrubbers. So all of these odor control set of equipment also have their own costs, right? And some of the bigger STPs also have hydrogen sulfide capture equipment, right? So that is very, very expensive. So the hidden, in terms of hidden costs, you have a significant increase in the capex of the plant. You have a significant fraction of the OpEx going to electricity. For example, for a 200 KLD plant, your electricity bill could be several lakhs per year. You also need backup diesel and a genset, which adds to the cost of operation. Further, as you have aging blowers and diffusers, they lose their efficiency and it necessitates new purchases. So basically what we have here is that we have an illusion of compliance. When you have equipment failure or media saturation, odor suddenly returns, right? That's a problem. So you have a sense of false comfort, right? Regulators and residents believe that the problem has been solved, but the root cause has been untouched. You have deferred maintenance. O&M contractors may sometimes fund the odor control repairs instead of the core uh, aeration fixes. As a result, the plant gets costlier, becomes more fragile, and still vulnerable to power loss, right, which is ubiquitous in India. So, what is the fatal flaw in the system? You have basically a situation where you're trying to make a resilient system, but that resilient system depends on continuous electricity. So, in the end, it's not resilient. You have power cuts that are quite common, which lead to a septic collapse. You have the odor control that you do becomes useless. The neighbors suffer, regulators may penalize you, and the plant fails. Ultimately, the truth is that expensive odor control solutions that mask the disease are not really the solution, right? There's nothing that is being done to treat the real root cause of the problem or to prevent it. So let us go back and see let us sort of understand from first principles. Um, is there some way in which we can generate oxygen biologically instead of mechanically via blowers? And the answer is yes. Nature has been doing this for a long time in all sorts of natural water bodies. For example, in lakes and ponds, you have algae and aquatic plants that photosynthesize during the day and release oxygen. You have oxidation ponds where you have algae and bacteria symbiosis that helps in oxygenation of the water body. You have no mechanical aeration. You have wetlands where plant roots release oxygen into the substrate, and microbial films help to oxidize organic matter. You have slow rivers where you have aquatic vegetation and algae creating photosynthetic oxygen and you know creating aerobic conditions for aquatic life to thrive and to maintain the water in a very clean way. So all of these natural processes, they maintain healthy DO levels without pumps, blowers, or electricity. So this is what is already happening in nature. So just to go a little bit more into nature and to understand what are the different plants that are there in a water body, and what are the important plants that can be studied further to gain some insights, right? So, like I mentioned earlier, you have plants and algal species that are ubiquitous in every water body. Bigger plants would typically sit on the lake bed, and there are many, many microscopic plants that are suspended throughout the water body. There are green algae, blue-green algae, diatoms. There are many, many other species as well that are just floating in the air, and they are microscopic, and they're very, very hard to see physically, but they can be seen in the microscope. When one very important photosynthetic organism is called the diatom. It is a single-celled algae with silica shells. It has a very high photosynthetic rate, and it multiplies many times a day. These algae consume nutrients during their growth, and when they do photosynthesis, they release oxygen in the form of nanobubbles for the aerobic bacteria in the neighborhood to thrive. The aerobic bacteria break down the organic matter, and when that organic matter breaks down, you have carbon dioxide released. That carbon dioxide is again absorbed by the diatoms, which release oxygen through the process of photosynthesis. The aerobic bacteria also outcompete the anaerobic bacteria and prevent the production of methane and smell causing compounds like hydrogen sulfide. So this is the base of what is actually happening in the aquatic ecosystem, right? This is the plants are the base, and these plants, specifically diatoms, they are consumed by another creature called zooplankton. When diatoms grow, the zooplankton population also grows in the water body. The zooplankton not only consumes the diatom, but it also consumes the mosquito larvae, the aerobic bacteria, and pathogenic bacteria, right? So the zooplankton is a filter feeder species. It consumes pretty much everything that is possible. Of course, it has its preferences, but it tries to consume everything and lets out the rest. So it consumes all of these single-celled algae called diatoms. And as the zooplankton levels grow, the fish also grow, right? So this in this process, the food chain is restored, and when you typically extract the fish from the water body, you have essentially taken out all the excess nutrients that entered the water body and resulted in the growth of diatoms, right? So the excess pollution into the water body initially gets into the diatoms, and then it gets sent up the food chain into the body of the fish and then out of the system, right? So that is how nature has always worked, right? Um, can we extract some lessons from this and see how we can take an understanding of this ecology into STP design, right? So that is basically what we are trying to do here in our company. We are basically proposing that you can have sewage entering, let's say, an STP where you have water collecting in a collection tank, and from the collection tank it comes into tank one. Some of the sewage settles naturally. The diatoms begin to grow, and as they photosynthesize, the bacteria begin to grow as well, and they get established along the walls. They get established on all the organic matter, and grow further, right? Because of the availability of food and because of the availability of oxygen. There's no electrical input required to make this happen. In terms of architecture, right? What is the architecture that we are proposing? We are proposing that you have a plug flow design where water comes into, say, cell one or tank one, and there's a high organic load. You have active bacterial colonization happening. You may have a little bit of denitrification if anoxic zones are present, but as soon as you have growing oxygen levels, you have excellent growth of aerobic bacteria and breakdown of the organic polluted matter. So you might have 3 or 4 cells. In the final cell, there will be what we call a polishing zone. You have some degree of clarity being established in the water body, and of course, some of the residual nutrients that are present in the water body are again recycled and consumed by diatoms for their growth. In this design, you have a continuous forward flow of water. Each molecule of water has the same residence time. There's no stagnation of the water body and no anaerobic conditions. So essentially, you're able to avoid those anaerobic conditions because there is photosynthetic growth of algal species that produces oxygen locally within the water body, within the tanks, naturally, right? In terms of design geometry, right? What are we thinking, right? To optimize photosynthesis, you can have different configurations. You can have long, narrow, or shallow channels, 1.5, 2 meters deep. You can have multiple, cells in parallel or in series. You can have baffles between cells to prevent short circuiting and force the sewage to contact the algal bacteria biofilm in each chamber. You want to have water flowing under the baffle so that you basically allow some of the solids to settle, and you force the water to spend the amount of time defined by the volume of the right. You want to have each cell has a certain retention time, right, defined by the amount of volume of that cell. So you want to ensure that water sort of goes in, um, comes down, and then goes back up and leaves the cell, right? So that ensures that water remains in the cell for as long as possible and doesn't exit the plant, right, very quickly without getting treated. So to help light penetration, you want to make the tanks relatively shallow. You don't have to make it very shallow because diatoms actually don't require that much light for photosynthesis. And if there is a limitation in terms of natural lighting, you can always put LEDs above the tanks or inside the tanks to ensure that there is always photosynthetic growth of diatoms. To retain the biomass, you have potentially you have a gravity fed sludge blanket is formed basically in the bottom of the tanks. You have biofilm support. You have because water flows from tank to tank through gravity flow. There is no need for pumps, no need for mechanical recirculation, and you just have natural flow of water from tank to tank. Essentially, what you have to do is to ensure that the inlet of tank 2 is slightly lower than the outlet of tank 1. And if you ensure that, then just by gravity, you will be able to make the water move from tank 1 to tank 2. So again, you repeat the process to ensure that water flows from tank 2 to tank 3 and tank 3 to tank 4 and so on and so forth. So, what do you need in terms of hydraulic retention time? 2 to 4 days would be optimum to get the best possible results. But even within a day of hydraulic retention time, you can get more than 70% reduction in various parameters. Um, the of course you have a trade-off in terms of footprint, land requirement, and treatment efficiency. The Of course, if you are okay with a slightly larger footprint, then you can actually do the treatment with no energy costs at all, right? So that's one of the trade-offs that are there. In certain cases, you can also adjust the hydraulic retention time seasonally. You might need lesser HRT in the summer when you have a lot of light. Maybe you can have a slightly longer retention time in the winter. But this is not very common. Typically, you can have one design that just works throughout the year and keep it simple. Of course, in terms of what are the trade-offs? In a typical deep activated sludge process tank, you might have tanks, especially in the municipal systems, that are 4 to 6 meters deep. Their goal is to drive high aeration efficiency with a compact footprint, but they're energy intensive, dark, and there's no photosynthesis or very little photosynthesis happening in those systems. For us, we want to have slightly shallower tanks, maybe 2 to 2.5 meters deep, or maybe even lesser. And if you have slightly shallower tanks, you might need to make them a little bit wider so that you have a higher surface area. This enables better penetration of sunlight and better photosynthetic growth of oxygen for a lower dosing, right, of the nutrients that you need to grow diatoms efficiently in the water body. So this is some of the trade-offs that are there. What are some of the additional features, right, of our FICO remediation-based STP? You would want to, ideally, sometimes in some situations, you want to have covered tanks, and you can do this. I mean, the requirement for covered tanks may come from a sense of aesthetics, or from a requirement to minimize smell. But even though you need open tanks to enable natural light to come in, you can compensate with LED lighting. And if there is LED lighting, you can essentially make the whole system closed. And when you make it closed, you can ensure that there's no odor escape whatsoever, even from the initial tank. As the water comes in, there's no odor. And of course, as the treatment goes on and the DO increases in the tanks, um, there's no odor in the final tanks, and especially even in the aeration tank, there wouldn't be any odor because the dissolved oxygen has been increased sufficiently. The advantages of the system is that it works, identically in rain, winter, dust seasons. It's weather independent, uh, and it has a very low power requirement compared to conventional STPs with blowers. The other feature I mentioned earlier is that you have gravity flow. You might be able to design the system with small level drops between tank to tank to allow for gravity flow from tank to tank without pumps. This system is resilient because even though you might have power cuts, the treatment continues because water is continuously moving through the system just by gravity. You have, like I mentioned earlier, you also have baffles in each cell to prevent short circuiting. And in terms of sludge, right? So there is a lot of benefit to the entire treatment process in this system because the dissolved oxygen levels in this system are very high and they are continuously present in all pockets of the water. You have very good ultimate treatment of the water body due to the growth of aerobic bacteria. There is very little sludge that is produced. The sludge that is produced, of course, will continuously get broken down by the oxygen that is being generated in the system naturally. So the amount of sludge that you generate in a day or in a month would be much less compared to conventional STPs. So this reduces the operational burdens and sludge disposal costs. So yeah, so that's what I wanted to say. And just to conclude, our belief is that sewage is not a inert waste; it is a living biological system, and if we start supporting biology and allow the nutrients to get upcycled, we can essentially make STPs that are silent, stable, invisible in some sense, and a tremendous resource for the society, right? Whether it's a resource for reuse applications in an urban context or for supporting aquatic life. If the water is let out into the rivers and oceans and the lakes, it can be a tremendous asset to the restoration of biodiversity and the aquatic ecosystems that follow it. Thank you. So I'll be happy to discuss further with any questions that you might have.
Thank you, Arun. That was a very lovely presentation.
Nikki?
Yeah, go on, Niranjan.
Mr. Arun Kumar, good presentation. Just want to know, in how many projects have you implemented this so far?
We have done, we have established, we have done several STPs we have typically, most often what we do is that we go into conventional STPs and we reduce the blower runtime and show that a continuous treatment based on diatom growth principles and phycoremediation can be achieved with little to no use of the blowers. So we've done 5 to 10 projects.
In Bangalore?
Not just in Bangalore, but in other cities as well. Tirupati, Mumbai, Delhi, Chennai, Bangalore.
Where in Bangalore?
There is one in Bommasandra, one in Hosur that is active right now. So,
Okay.
yeah.
And did I hear you right when you said that you upgrade existing STPs?
Yes, that's right. That's right.
Okay.
So yeah, what I talked about today is more of a first principles design where we would design the entire system without much of the equipment that is conventionally found in an STP. But most of our current projects or prior projects have been retrofitting older STPs.
I just want a general figure. What will be the approximate cost for upgrading an existing STP, which is about, say, 300 KL?
Yeah, maybe we can discuss this offline.
I just
But
want a general. I don't want you to, I'm not going to hold you to the
But
price.
see, what we do is, we have the capex that we add to the system is almost zero,
Okay.
and we can treat the water for maybe two rupees per kiloliter or less.
Okay. Thank you.
Yeah.
Uh, Arun, one, if I can request you to stop sharing screen.
Yeah.
The
Uh,
second is there have been you've been witness to a lot of very lively conversations in our group also on the efficacy of such a system.
Yeah.
Um, and by first principles itself, I mean, I'm going to request other people, if Snehilji is here and Sanjayji, and who've been working on STPs, large scale STPs, to kind of come in and see how how this is. I mean,
yes.
we were part of a conversation with Ravilji also kind of, questioning the first principles of such a system. And if you can tell us a little about, the results that you've gotten of treated water, as well as how the treated water can further be treated for health grade water.
Uh,
Yeah. Neil, were you saying something?
can I ask some questions?
Yeah. I mean, let me just
Answer. Okay.
just yeah, go ahead quickly. Just answer to Nikki's question. Absolutely, I think see the discussion of the results. I'll be happy to share with you or anybody the results that we have achieved in the previous projects. So a few of them are municipal STPs. Some of them are apartment STPs. Some of them are industrial STPs. Most of them are retrofits, and we've shown dramatic reduction in the blower runtime, a reduction in sludge, high levels of dissolved oxygen in the output, and achieving PCB mandated parameters while doing all this, right? The In terms of first principles, I think I kind of alluded to the principles behind our treatment. We are seeking to, we deploy our nanotechnology products to accelerate the growth of diatoms in these, starting from the collection tank of an STP. And this results in early start to the treatment process in a conventional STP, longer retention times, higher oxygen levels, enabling better breakdown of the organic matter, and also better. And because you have an algal bacteria symbiotic relationship, many of the bacteria that die and are remineralized, they are reabsorbed by the diatoms. And so the minerals are reabsorbed by the diatoms. So there is less sludge in the conventional sense being produced in our system. So, Sanjay, you had some remarks?
Hi, Arun. Very interesting. Now, I'm going to ask you from issues that I am facing. Okay, we've been putting nature-based solutions from villages to factories to other places. Uh,
Mhm.
our system basically has anaerobic biodigesters at source. Then we only transport the liquid to the STP. Okay, so
Sure.
now I am looking at options for STPs. Currently, we are using that VEC Camus SBT.
Mhm.
Yours looks like an interesting solution as well. Now, I have a couple of questions. The parameters you'll reduce would be BOD, COD, TSS, nitrates, all the standards
Phosphate.
reducing.
Yeah, try nitrates, phosphates,
Yeah.
sodium. So because we are relying on algal species, right? So like
Yeah.
algal species, like any plants, they absorb all the nutrients that are there.
Okay.
Uh, and
So, second question, you need sunlight. Good sunlight
yes,
would be preferable.
sunlight or artificial light, sunlight or artificial light. Yeah.
And temperature
There is as long as the water is not above, I'd say 40, 45 degrees, you're
and
good to
below,
go.
bottom.
Bottom, there's no even 4 degrees, 5 degrees, 10 degrees. C is fine, no
Okay,
problem.
because see, I've got a project in Ladakh,
Yeah.
which hopefully will be starting in Leh area. Hopefully, April, May, we will start it. Uh,
Hmm.
so I'm looking for a solution that would work in those conditions. So I
Yeah.
can put the system in a greenhouse so that there's no snow and all covering it.
Yeah.
But will it work in those kind of conditions? There's plenty of bright sunlight. That's not a
Yeah,
problem.
see, yeah, yeah. So, see, diatoms are known to grow even under the ice sheets under the
Okay.
Arctic, right? So,
Yeah.
diatoms are a major portion of the food chain in
Yeah.
oceans, and they
Okay.
survive below the Arctic ice, where
Okay.
fish are still found.
Okay.
And so, we are confident that diatoms grow at very, very low temperatures, and as long as you can grow diatoms, then you're good to go.
Okay. Do you need, what happens with things like chemicals? Suppose they're using harpic acid to clean the bathrooms and all that. While
Uh,
diluted, it will come into the system.
yes, see, there are certain chemicals that might retard the growth of diatoms, like certain pesticides and sometimes phenolics.
Yeah.
And so, as long as the concentration of those compounds are not too high, then it should be fine.
You
But
won't
it
find
needs
them
to be
normal.
checked.
You won't find them in normal household usage, no?
Yeah,
Those.
but yeah, I think see, we have operated this in apartment STPs,
Yeah.
and there's no issue. So unless you have some big industry or something that is really making things really bad, right? Or
Okay.
by intention somebody is trying to sabotage you, then it should be
Yeah.
fine.
Okay. Then one last question I have. In terms of uh, area requirement, uh,
Yeah,
so you said 1.5 meter long channels, 1.5 meter deep channels.
yeah, one and a
Would
half, 2 meters. I think I tried not to go more than 2.5 meters deep, but
no, 1.5 is fine. We want shallower.
yeah,
We would
shallower the better. Yeah, yeah,
yeah. So if you have, let's take a number. Let's take a 50 KLD unit.
yeah,
Suppose I give you BOD around 60, 70, COD maybe 100, and
yeah,
TSS maybe similar, something similar, 50, 60 or something like that.
yeah.
What kind of space requirement would be required in terms of these channels and all? I'm trying to figure that out because I need to think about that as well.
Yeah, sure. See, we nominally suggest 2 days of retention time.
Yeah,
So 2 to 4 days is longer the better.
yeah,
And depending on the input water quality, you may need more or less. So some degree of trial and error is required.
yeah,
If you make the tanks in some sort of a modular way, then you can
yeah.
add tanks to get the water quality that you need, right? Uh,
Okay, so there's a series of parallel ones. One will overflow into the next one. It'll
that's
go
right.
from underneath, from under the baffle, like you said. So it'll
Yes.
go
So
from
so each tank would have a baffle wall in the middle, so
yeah,
that water entering it from the top would go
yeah.
under the baffle and come up. And then
Okay,
as it exits, the next tank will be at a slightly lower level, so
yeah.
that it goes in there, maybe 2 inches lower than the first tank, so that and that cell will also have its own baffle wall in the middle, and then water will sort of go down and come back up.
Okay.
So, you know,
Fair enough. And end product will be under those current MPCV norms, 20 BOD,
Yes, absolutely. Yeah.
all that sort of 50 COD and all that sort of thing.
Yes, 50, 50, 10, 10, 10,
Yeah.
1. Yeah.
Okay. One is what? Nitrates.
Phosphates. Yeah.
Phosphates. Okay. And nitrates?
10.
10. Okay. Fair enough. Thanks. That's my question.
Yeah.
Thanks.
Thank you, Sanjay. I request Neil to go next and
Yeah.
then Ajay Singh.
Hello.
Yeah.
Hello.
Hello, Neil.
Yeah, I'd request Neil to go next.
Arunji?
Just a moment.
Hello.
Arunji?
Can I ask?
Go ahead,
Yes.
Neil. Go.
Okay. Okay, proceed, sir.
Arun, good evening. Actually, I couldn't join as somebody was discussing water problem with me, and he was not leaving me. Because of that, I joined little late. I will look forward for the recording. Anyway, I have two questions. The guy who came to discuss was asking about a six house rental thing. He is after constructing. He is struggling to balance the water requirement.
Okay.
So his question to me was, I told him repeatedly, I have a seminar on the same, was that how can he has made the water line into 3-4 lines. One is rainwater line, one is grey water without kitchen, and kitchen line, then black water line. So he is asking, can we recycle the grey water and use it for flushing? Okay, because he did the measurement of the houses. He is finding daily he need 2000 liter for the regular water and 1000 liter for flushing alone. So can we make with this thing? That is the
So
question I want to
are you proposing to treat only the gray water?
Yes, grey water.
Okay. See, we typically don't recommend usage of this approach for only gray water. And that's because, for diatoms to grow, you need a certain ratio of carbon to nitrogen to phosphorus. And typically, the gray water may not have as much carbon sources. We would prefer that the water be mixed with, black water, every kind of water mixed together, right? So that you have all the different nutrients that are required for the sustained growth of diatoms. I think Bhaskar has also looked into this quite deeply, and perhaps he can offer some clarifications or illuminate.
Okay. That is can that Bomasandra project can we visit? Is it possible? I am near to that.
Yeah, sure. I can make a request and you can come and see.
So if we get your number, I can talk to you.
Sure.
Second thing is, like, I am longing for doing a project in my home where unfortunately my friend who built the house was not ready to separate the black gray waters together. So I want to mix, I want to have a solution where I can recycle this water and if possible use it for flushing, otherwise for plants thing. So a house level, our consumption daily is 500 liter max.
Yeah.
For the house. Okay,
Yeah.
5 members, 500 liter max. Is there any way we can do a model project? Because why I do think about is these individual houses in Bangalore or across India. There's for a little bit of saving of plumbing. We are mixing black, gray, and everything together.
Yeah.
In this condition, if one time government also comes or people become bothered about it, there is a high need of recycling this water and using it. So I want to spend, even I can spend, and I want to do a model project to make it happen. Is that possible? Can you help me out?
Yeah, I'll be happy to help you with a model. We should do it at some scale. We should try to, slightly bigger. The bigger, the better. Because sometimes when you have too little bit of water, then you may not have enough dissolved carbon dioxide to sustain the growth of diatoms. So I would always suggest that we do bigger pools of water because, that, the
So
raw.
what is the minimum in your methodology you are suggesting?
I would do several kiloliters per day minimum.
So several kiloliters. That means
Yeah.
even 2 or 3 is also not enough. probably something like 10 kiloliter.
Yeah, I think 5 to 10 is what I would feel comfortable with. Yeah.
Okay.
Yeah, I mean,
Anyway,
we haven't experimented at that scale. So to be honest, I don't know. We have
so,
always done
like
30,
in
40,
person,
20.
we will
Huh?
interact. We will interact personally and try to
Yeah,
see,
yeah.
like if there is a possibility, because I want to make a model project and I know for sure it is going to happen.
Okay.
Okay, even at the individual house levels. And you also please explore the way of modelizing and trying it out. You
Yeah.
can use my house,
Okay.
for the thing, right?
Yeah, I'd be happy to discuss with you and work with you on
Right.
productizing smaller ones.
Yeah.
Thank you.
Thank you very much.
Hello, sir. Hello.
Yes, hello.
Good evening, everyone. Yeah.
Hello.
Yeah, we can hear you, Narendra. Please. Go ahead.
Arunji,
Yes.
can I?
Go ahead.
Hello.
Yes, go ahead.
Yeah, this your principle of working is okay, but that degradation by that diatoms, etc. Thing is that what is the per meter square? How much COD or BOD it can reduce your plant? First question. Suppose you have a plant of say 10 square meters plant. I mean, your tank is there. How many kgs of how many grams of COD you can reduce per day? What I have doubt in this subject is that in your subject, yes, it will work like any other treatment plant, but the rate of reduction, rate of degradation is very less compared to the forced air, forced oxidation with the air. By growing the more and more bacteria and microbial this thing and degrading, you require very less area, whereas in your case you require more area. In fact, our more concern in any urban area is that land is the main problem.
Yeah.
To construct a treatment plant with the existing forced treatment, also people are not finding sufficient land for the treatment plant to have for each colony or each cluster.
Yes.
So in this scenario, how efficient your system can reduce the area?
See, we can also think about potentially doing a vertical cascade of tanks in our system. So if you can go high, you can definitely reduce the footprint even more. So there's
can I have some idea of the structure of that high, how it will be? You are covering the water surface with your plantation. Am I right? That is what you call.
No. So the idea is that we would put certain nutrients, nanonutrients into the water.
Agree.
And that will support the growth of plants, diatoms inside the water.
Yeah, these diatoms will be there on the surface or at the bottom?
No, it'll be there throughout the water body. It'll be there it'll be spread throughout the water.
In my experience, if the bottom, suppose any solids are there, precipitated matters are there, they go and settle at the bottom. In course of time, they form the anaerobic bacteria there, and it will start anaerobic digestion. When anaerobic digestion takes place, and this aerobic always going for problem.
Yeah.
So unless there is excess area, oxygen present in the system,
yes,
the anaerobic cannot be stopped.
Yes.
How do you create excess oxygen?
So the way we create excess oxygen
for the material balance, how do you create the excess oxygen? Whatever it is organic is taken by the oxygen generated by your diatom.
But we are generating much more oxygen. We are generating much more oxygen. The diatoms are growing rapidly.
So, what is the chemical structure of the? Can I feel the chemically? What is the composition of your diatom?
No, so the diatom
is elemental.
So,
what elements?
Yeah, we are not adding diatoms to the water body. The diatoms are naturally present. What we are doing in our approach is to add nutrients to accelerate their growth. And we do that continuously. So these nutrients are added with a dosing station next to the collection tank. And some small amount of our nanonutrients are added to the collection tank 24/7. And they will sustain the growth of diatoms in the collection tank. And then since that water moves from tank to tank, it will sustain the growth of diatoms in all the tanks as long as there is light present. So as long as you have LED or a natural light present, the diatoms will continue to grow and continue to generate oxygen. And the oxygen that is generated is interestingly in the form of nanobubbles. So they have a very long residence time in the water. So they don't bubble out like conventional aerators. So the water will just be there. It'll be a very quiet system, but it'll be fully saturated with oxygen. And you can take a dissolved oxygen meter with a probe and you can dip it at the surface. You will measure a certain oxygen level. You can dip it further. You will still measure some oxygen. If you go further down to the bottom, you will still get enough oxygen to avoid anaerobic zones from getting established.
Okay. My first question, please, if it is there, the data with you, for 1 square meter of your water depth, whatever it is there, 2 meter, 3 meter, what you said is we'll take it. How much of COD can be reduced for this per square meter of the tank?
Yeah, I don't have that number off the top of my head, but I will calculate it.
No, at least you tell me what is your experimental your experiment you have done with these tanks. What is the size of the tank? How much loading you are doing? What is the COD input and what is the output to COD?
I think it should be taken up. Narendra Garu, you should take it up one-on-one with Sri Arun instead of taking all our time with a specific answer, because it's individual consulting.
Thank you.
Okay. This is Raj from Chicago. I have a quick question, Arun.
Yes, hi Raj.
Yeah, this nutrients that you add, how much does it add to the operating cost of the plant? And is it offset much more by the
It doesn't add to the operating cost
saving
of the plant. It more than offsets the reduction in aeration and sludge disposal costs. So you will have a net savings quite easily. So I think
this is fantastic approach, because in Chicago we have huge blowers and aeration tanks, and because we deal with almost 900 billion gallons of water during heavy rain periods in our sewage system. But for smaller systems, this is an excellent approach, and I think if you can publish some papers internationally and get this information known about your nutrient addition and how it offsets the amount of oxygen that is required through the blowers and all that, it'll be a real benefit to society worldwide. Okay, thank you.
Yeah, sure. Thank you.
Wonderful job.
Yeah, we do have a US patent for our product, and there are a few publications. I'll be happy to share them with you. And of course, a lot more work is required to really take this and get it completely accepted into the various institutions around the world. So
Nikki has my email. He can give it to you, and if you can share it, I'll share it with some of my people I know at the Water Reclamation District in Chicago. Okay.
Okay. Thank you.
Thank you.
I'll share contact with us.
Thank you.
Ajayji, you had something to say.
Okay. Yeah. I think there's some cross connection. So my question was, there is some biomass in the tanks of diatoms itself. When you talked about diatoms initially, it's part of a food chain.
Yep.
So that kind of diatoms get eaten up. But what happens to this? How this biomass is tackled? Because the diatoms have a age, and after they do their business, that it will be sinking down to the tank.
Yeah.
So this will keep on accumulating.
So the diatoms, most of them diatoms will be consumed by zooplankton, and whatever is not consumed, most of them will either flow out with the water. Either the diatoms will flow out or the zooplankton will flow out. In either case, there won't be too much diatom accumulation within a conventional 1 day, 2 day time period. Diatoms have a much longer lifetime, around 5 to 7 days minimum. So you don't expect diatom death. Of course, bacteria will multiply, bacteria will die, but some of those nutrients released by the breakdown of bacteria will get remineralized and reabsorbed by the diatoms. So therefore, the accumulation of bacteria as part of the sludge is also reduced.
Okay.
Yeah.
Thank you.
And of course, because you have a higher dissolved oxygen level, you are able to actually break down the organic matter more completely. So even typically sludge is in a conventional system, sludge is a combination of biomass and unbroken organic matter. If you break down the organic matter more completely, then you have less of it available. And if you remineralize the broken down, the broken down bacteria and reabsorb it into the diatom, then you will have less of that also available. So the end result is that you have less sludge accumulation.
So how do you do some kind of process control there, because you are dosing?
Yeah.
And there's a certain amount of
Yeah, typically
diacetyl.
you would control the process by measuring dissolved oxygen through through handheld meters or through an automated meter,
So
and you
it's
would
like
maintain.
it will go up and saturate, and then it will start dropping basically after a certain
No,
point.
dissolved oxygen will stay steady because you have oxygen coming in and oxygen, you have sewage coming in all the time, and you have nanonutrients coming in all the time. So a
Mhm.
steady state is established within a couple of weeks or so, right? So we have a commissioning period during which there are all these changes happening. So in a
Mhm.
2 to 4 week commissioning period, you can stabilize the dosing scheme for the kind of input loads you have, for the kind of input water quality that you have, and so forth, right? So you have
Yeah.
to spend a few weeks commissioning the system. Yeah.
One last question from my side is that it's dependent on sunlight. So there's some element of seasonality to it, and in those geographies where you are having more cloudy days, the function might not. I mean, it might not work during those times to its full capacity. So that means LED lights are kind of built into the system.
Yes. We normally build in LED lights, to ensure that you have diatom growth that happens 24/7,
Yeah. So
and to compensate for any fluctuations in the natural lighting.
okay.
Having said that, diatoms actually require very little light to do photosynthesis, and it is for that reason that they grow quite deep in water bodies as well, right? And compared to green algae, which require much more light, to do
Mhm.
photosynthesis. So green algae tends to grow closer to the top, but diatoms require less light intensity to do photosynthesis, and therefore is able to grow at deeper depths. So there is a certain amount of resilience that is built into the system, because we are utilizing diatoms to accelerate nutrient absorption and oxygen production, right? So it's a good species to work with.
Okay, you have tamed it.
Yeah. Yeah, so there is a company in the US that is working on green algae based, algal based STPs, right? So yeah. Yeah. And
All
that's a
right.
different
That's
design
all from
philosophy.
my side.
Huh?
Thanks.
That's all from my side. Yeah.
Yeah.
May I ask now?
Yes, go ahead.
Yeah. Basically, the thing is that when you say that oxygen is taken by bacteria, because you are showing here that the bacteria is aerobic and they take oxygen. Is it O2 oxygen, or which oxygen does bacteria take? That is first question. And secondly, when you say that the sludge gets reduced and diatom plays the role and sludge gets reduced, did you analyze the water before going for the treatment for the ratio of eukaryotes to prokaryotes. Because in eukaryotes, when I say, that means there is a lot of organisms which has chitin, which doesn't get dissolved, or say doesn't get broken even for a long time. And that is how when we see the sludge is more in the system of conventional, it is because of these kind of things only, which are outnumbering the prokaryotes. Next is about, you said that you are trying to say that there are some new nanonutrients which is being provided, and then diatoms, and then again you were talking about the zooplanktons which keeps it in balance, while diatoms and zooplanktons, they are in the system, and then they keep a balance of how they eat away, or zooplankton eat away the diatoms. And again you say that the bacteria, when they die, they will be eaten by the diatoms, and then how it's like you are talking about the remineralization and other things. So, what is the claim that you are doing on the basis? Because as we understand, it's all about the redox potential. And initially, when you started with the even odor, when you said that it's that bad odor which is there, but odor means itself that there is some that there's a oxidizing which is happening, and its main thing is about the pH, which is able to make the system in such a manner that where these bacteria are able to function. So it is mainly when we talk about biological degradation, we talk about how microbes work in the system. You are saying more about the diatoms, and you said how diatoms are in the balance with the bacteria. So first question of mine is that whether bacteria takes O2, oxygen, which you say, that is dissolved oxygen, when you are saying that oxygen level is high all the time. So I just wanted first answer to that.
So my understanding is that it is just O2. Bacteria do take the O2 in that form.
See, it is always that the hydroxyl and the hydrogen ion, which is getting broken while the breakdown is happening. If you take an example of pyruvate to citrate and isocitrate, in that process, if you have seen the TCA cycle and the reverse TCA cycle and all, you know that hydroxyl and hydrogen ion, when they break up, and in that case only rearrangements which is there, that is the intracellular intramolecular rearrangements which is there, which we call it as the redox chemistry. So in that process, whatever the oxygen which is released and again taken back, in that process, whatever the oxygen, when we talk about the anoxic world and the oxic world, from anoxic world it came to oxic world. So from that point of view, I'm trying to say that when we talk about the cyanobacteria and others, from their poop only the oxygen came into the world, and then that's how oxic world started. And here you are trying to say that bacteria takes O2.
Aerobic, as far as I know, the aerobic bacteria, they oxidize organic food through their electron transport chain, passing electrons until they reach O2 at the end, right?
Yes,
O2 accepts these electrons and combines it with protons to form
but
water,
then
closing the
that is
respiration cycle and generating ATP, right?
Yeah, but then that is the anaerobic process. See, that's what there is a confusion between an aerobic and anaerobic process. Now we know that the bacteria's nature is also facultative. Like, under stress, they work very well. They can easily switch on and switch off to aerobic or anaerobic conditions. That is the nature of the bacteria. So that's how they function. But when we say that oxygen, O2 is not the oxygen which is being taken, that atmosphere. That's why we are all against to that atmospheric blower which is happening, or the aeration which is being done in the conventional system. That is totally wrong, because O2 is not being taken by the bacteria. It's only this process of rearrangement, intracellular, intramolecular rearrangement which happens, and in that hydroxyl and hydrogen ion, when they separate and then again they join, that means when you say from pyruvate to citrate, citrate to isocitrate, and cis-conatate. So in that process, you will see that the breakdown when it happens, and in that the release of oxygen which happens, and again it is taken back at the next level. So that give and take of the oxygen which is happening there, and that oxygen is the main which is being used by the bacteria, not as an atmospheric or the DO which we are talking here. DO is totally different kind of an oxygen, and here we are talking about the aerobic and anaerobic process of bacteria. So there I feel there is some confusion. And basically when you talk about sludge, that also did not go well with me, because I believe that sludge, when you say it has a lot of you initially, that is the reason when the sewage comes into the system, it's very, very important that we have to check that how much amount or the ratio of eukaryotes to prokaryotes and all that. And today in the conventional system or any of the sewage, when we see, because of the long conveyance system, that means the layout pipeline from by transportation by which the water is brought to the system, and in the process a lot of eukaryotes come, and these eukaryotes are more with these kind of chitins and all, which is very hard to digest or say to break down, and this is what is creating a lot of sludge generation. And here you say that there is no sludge generation, and I hope you know Preeti Rao. I had spoken to her also, and what you were talking right now at a place where you have done this particular work. Of that, she has shared some work and even your research paper, where I found that there is no, no analysis with a molecular lens, and that is much more needed. We in everywhere, whenever we are talking, it's like yes, COD is coming down, BOD is coming down, or phosphate level is this, that, but what is causing this, how it is happening, what is the science behind it? Nobody is still talking about it, neither anybody is explaining. Right now also somebody asked about the principle behind what you are doing. At that time also I see that you have spoken about some principles, it wasn't more like a science, but then that these things are being handled, that things are being handled, and then nanotechnology or with the nano some food nutrient which you are providing, and with that whatever the diatoms are growing, and long RT and high oxygen level and all that you have explained just now. But here again I repeat that bacteria don't take oxygen like us. Bacteria takes the oxygen which is in the process of breaking down, that is redox chemistry, the oxidation reduction reactions. So what will you talk about that?
See, I think, my understanding is relatively simple, right? I think we need, as far as I understand, diatoms release O2 when they do photosynthesis. And that O2
I'm
is
not
absorbed
talking
by
about
anaerobic
diatom.
bacteria.
I'm not talking about diatom right now. I'm talking only about the bacteria, because it is all about the bacterial degradation which we are talking about in the process when we talk about the
Right, but
organic matter.
that O2 is coming from the diatom, right? So the O2 is coming from the diatom, and so that is important. And so,
That is important for the phytoplankton, zooplanktons, animals to survive in the system. Here
yes.
we are talking about the degradation of organic matter, and we are talking about pathogen elimination. So from that point of view, that we have to discuss here, because most difficult task or today, if in Prayagraj or anywhere, because I was a bit confused when you were talking about that photosynthesis and all. This is when you are talking about lake system like treatment, and when we are talking about purely the sewage treatment and all, here we don't have to talk about photosynthesis and all. Of course you are talking about treating with diatom, so you are talking about now photosynthesis.
Yeah, so
So
so these are completely different treatment philosophies, Snehal.
yes, yeah,
We have a very different treatment philosophy, and the
yeah.
anaerobic community has a different treatment philosophy and a different process, right? So there are situations where somebody might want to use our approach, and there are situations where somebody might want to use that approach. So we have no argument there, right? So I think we should keep the two systems separately, and each system has its own place in the world. That's all.
Yeah, but then here we are not talking minus bacteria, no, we are including bacteria while we
Yeah,
are
yeah,
talking about the
yeah, yeah. We're
treatment.
talking about
So when
aerobic
we are,
bacteria. That is the
yes,
only
yes.
bacteria which I know about, right? So,
Yeah, so when you know about the aerobic bacteria, you have to understand first what kind of oxygen does the bacteria take. And when we say what is aerobic and what is anaerobic, when we talk about the bacteria, what is that situation? And now when you were talking about the gradient level also, I was hearing that you were saying as you go deeper, your oxygen level gets more and more.
no,
And
no, no, no.
even
I'm not saying that. I'm saying that the oxygen level as you go deeper is, I'm sure it is slightly lesser than it is at the surface, but it is higher than what is required to get out of the anaerobic condition. That's all.
Yeah, but still the question remains the same, that here when we talk about the degradation, is it the diatom doing the degradation of organic matter or your bacteria? Because
Bacteria.
here
It's
you are,
absolutely bacteria. Bacteria
yes,
is
so
doing the job.
yes,
See,
yes.
I
So
think, I'm Dr. Srinivasan here. See, one of the things that you need to understand is probably what where Dr. Arun Kumar is coming from, is that you provide an environment which is aerobic in nature, right? Where aerobic bacteria can thrive and they do their job. Now, if you don't provide dissolved oxygen, then obviously you're going to have anaerobic bacteria which grow in. So all we are doing here, he's probably saying, is that you provide an environment which actually allows, fosters the growth of aerobic bacteria, and they do their job. The exact mechanism of how exactly these aerobic bacteria take in the oxygen, or how do they work, that's probably for academicians like you to actually, do research and find out.
no, but
This is a
you are
system that he
arguing the
claims he
aerobic.
works, right?
You are arguing the aerobic with the O2 which is being released by the diatoms. And you are saying
Absolutely,
that that is
absolutely,
the food for this aerobic. And we are
absolutely.
trying to say that O2 is not the thing which is being taken by the bacteria, but it
It
is
may not
the
be. No,
oxygen
but the
only
point
by
is you are
the
providing
intramolecular
but if,
arrangements which is there.
no. But if oxygen is not there and is not dissolved in the water, obviously aerobic bacteria will not survive. You need
Why?
an aerobic. Why? How
That's
do you
what
think
I'm
that
saying,
aerobic
that
bacteria would survive in an?
aerobic bacteria doesn't means that there is O2 or dissolved oxygen in the water system. So
But
that we need
aerobic
to
bacteria do survive in a system where there is dissolved oxygen, and when there is no dissolved oxygen in the water, you do find putrefaction. You do find hydrogen sulfide and other gases being emanated, and you get foul smell. What does that
see
mean?
just now I
I
said
think I
there is
disagree with you
no
on this
there is
point. You need to understand that maybe you're talking about
bacteria
the.
keeps evolving. Bacteria keeps
That's all
evolving.
fine. That's all fine. But yeah,
That
that's all
is
fine. But
already
I think
been
we should take
proved
this into a
by the
different kind
scientific
of a
community.
discussion. I think we are
No,
holding back all the others also. Please hold on.
Snehal and Srinivasji, I mean, this is a very interesting conversation, but I would, I mean, this has been a long-standing conversation. I think we need to have like a
Yeah,
series
I think
on
we
that.
need to allow others to,
Yeah.
bring their points into.
Yes. I
And
come
by the
from
way,
the
I'm a
field side.
doctorate in
I come
chemical
from the
engineering,
field
so I do
side.
come from, right.
Rather than from the lab side, I come from the field side. So I was
May I
listening
know
to the.
Dr. Sneha's background, please?
Yes, hello. Yeah,
Go on,
yeah,
Snehalji.
yeah.
Dr. Sneha,
So,
please.
yes, so I have two PhDs, third going on. First
Okay.
PhD was in toxicology, second one is in management studies, third is going on in Hinduism and social sciences, that is ecology, which I'm doing on Ganges. And my basic my profile was I was for 20 years degree college principal in Mumbai University affiliated college. Again, I was given a vice chancellorship, but I denied for that because I wanted to work for community. And since a long time, almost 2 decades, I am working for the river rejuvenation initiatives across India as well as abroad. And recently, I have come back from Fulbright Scholar in Residence from Manhattan City's 286 million gallon capacity sewage treatment. They don't call it as sewage treatment plant, but they call it as water resource recovery facility. I conducted complete study and a global review of all the STPs which are put, and then a report has been submitted to the New York governor. So that is what the background I bring it with me. And then a lot of like even my proposal has been accepted by DST as a postdoc women scientist for 3 years. And the same on the same topic, I'm trying to give an alternative to the sewage treatment. And I mostly I propagate for nature-based strategies rather than
Then
nature-based.
you must probably you encourage people like Dr. Arun Kumar,
Yes, absolutely,
instead of finding
absolutely, absolutely. And even
gaps
I
in
am
his
a
thing.
chairman.
Yeah.
Even I am a
Srinivasji,
chairman of.
Snehalji has been doing
One
some
thing I
very,
will
very
just
good.
add here, just I will add here. I am the chairman of Clean and Healthy River Committee, Microbiological Society of India. So I bring in that expertise also with me. So that is why these questions were. Thank you.
Thank you for your
Yeah,
inputs.
I'm
Yeah.
waiting for my turn.
Yeah.
So this is
Go on, go
Gautam.
on, Gautam.
Yeah. So I come from a field rather than the lab side. So rather than from an expertise point of view, from a general layman point of view, how this system, I gone through the conversations. How is this different from a broader segment of DVATs, the system you have proposed?
See, DWATS is, as far as I know, is basically a sludge of. I think it is a controlled plants, right? And I think it's mostly an anaerobic process, right?
Yeah, it's there is a lot of anaerobic is involved and canalo plantation is involved.
Yeah. See, I mean, as far as I understand, DWATS is anaerobic first, and then an aerobic MBBR or a trickling filter, right? It relies on gravity flow and with very limited mechanical aeration, right? So, I mean, let's see. I think in a
Because when I
DWATS,
saw the title of the presentation, the first thing that struck in my mind was about DVATs only.
yeah. See, DWATS is anaerobic first. In our case, we are completely aerobic from the beginning, right? So we have photosynthetic growth of diatoms and the bacteria rely on it and so on and so forth, right? So if you want to know about other differences, for example, in DWATS, you still do need some pumps and fans, whereas in our approach, you don't need that as long as you have enough retention time and good flow. The DBAT system is compact modular tanks, right? It's good for urban sites. In our case, we use existing tanks and so forth. If you have an existing STP, we can just work within that system.
So have you studied DVATs and address any of the drawbacks in
So
the
the divides, the biggest divides is sludge, right? Because from your more sludge is generated from anaerobic digestion, right? And it also needs dewatering. In our case, you have less sludge, and you have aerobic digestion and cycling of algae, right? So no, I haven't studied in too depth, but these are a few basic points that I can make, right? So most of the DBAT systems, they meet the discharge limits, but they may need polishing, right, for further reuse.
So as per your understanding, like from a commercial point of view, how we see is that if you in the urban areas, I think the most sensitive subject is the real estate.
Yes.
But when you go the interiors, like where the real estate is not a big issue, there the concern is OPEX should be as minimum as possible when the land is available.
Yeah.
So people, I mean, there I've seen a lot of. In fact, I myself have installed for an education residential institution like a 70 KLD DVAT system. Just trying to figure out how your system is a bit different from DVATs. It is actually designed by Indumati Nambi, IT Madras professor.
Okay. Yeah, like I said, it starts with, I think we would have to really do a more detailed analysis of CapEx, OpEx, footprint, energy, sludge, right? So these are the five different vectors in which the two systems need to be evaluated and, compared.
Sorry,
And
CapEx, OpEx, and what is the other parameter?
sludge generation.
Okay, sludge generation.
Energy utilization.
Yeah, I think that comes under OpEx.
Yeah. Sure.
All right.
Yeah,
I think
and then suitability for rural or urban contexts. That is also something to be studied.
Yeah.
So I think we can address this offline. We can have more detailed.
Yes, that's what. How can I get your number?
It is there in the chat.
All right. Thank you.
May I go ahead?
Yes, go ahead.
Go ahead.
Yeah. So you mentioned about retention time of 2 to 4 days, and also mentioned that in Bangalore you had retrofitted in the existing
Yep.
Standard systems which are there, right, with the blowers and
Yep.
stuff like that. So there, the retention time is lesser than 24 hours. So how do you kind of, if a system is there, the retention time is less?
One of them is a 24 hours kind of retention time system, and we run it with just 2 to 4 hours of aeration compared to what it was earlier. There is another STP which had, in fact, it is completely oversized. It had few days of retention time. And there we just shut down the blowers and we just allowed the water to go from tank to tank. It was not gravity fed design, but there the water was being pumped out from tank to tank because that's just how the system was.
Right. So essentially, what I understand is if the retention time, because of the design limitation, is less than, let's say, 24 hours, then it can be augmented using the blowers.
Yeah, blowers do achieve do a little bit in terms of mixing the water a little bit. So there is some function to that. We don't need the blowers for producing oxygen, but just for creating a little bit of mixing effect. And there
Got
is a benefit to that.
And to the point that you said, if the system has pumping from one sort of a cell to another cell, in that case, does that impact the diatoms in any sense?
No. There's no impact on the diatoms at all. It just moves with the water.
And in the situation, let's say there's so you are in Bangalore, but let's say in other places, if some place already has a standard STP and they would want to kind of try out or explore, what would you recommend as a process? What do you
Yeah,
typically go as
sure.
for?
See, there is a great variation in STP design and process methodologies, right? So we would need to you would need to have a discussion about the specifics and then suggest what could be done to help that particular STP operate better.
So, no, so what I'm trying to understand is broadly what I understand from the discussion is primarily it's all about based on the whatever existing parameters, energy consumption, the size of the tank and everything, you would be deciding what sort of dosage would be required,
Yes,
and then
yes.
dosages is what will be done by the team over who is operating the STP.
Yes. So they just have to put some light bulbs above the tank. So I will specify what wattage bulb you need to put above and how many bulbs you need to put above each tank. And I will suggest how many milliliters or per day you need to dose, and where you should establish the dosing station, and then we would just continue with your existing process.
Right. So essentially, you don't need to physically attend or see or inspect the STPs for that purpose.
No. But we do need somebody who is ready to sort of work and really observe the oxygen levels, keep track of the oxygen levels, ensure that the correct MLSS is maintained, ensure that the correct oxygen levels are maintained, and uh, proportionally turn down the blowers so that the treatment process is as efficient as possible.
And in this whole process, just a possibility that people keep thinking, what could be the worst case thing? I mean to say, just in case things don't work. Is there
See,
anything?
you can always return back to your original situation, right? So I think it's important not to overdose with our nanonutrients, and you don't want to increase the DO levels too much because that might having a dissolved oxygen above 8 or 10 can sometimes promote the growth of filamentous bacteria, which is not healthy. So we, you know, ask people to ensure that the dissolved oxygen in the treated water is around 6 milligrams per liter. And just ensure that if the inflow is less, then proportionally turn down the dosing. If the inflow is more, you proportionally increase the dosing a little bit, but don't exceed your dosing levels. Right?
So it is all based on the key parameter to control is
Oxygen.
the oxygen
Yeah.
level.
Yeah. So,
And
so to control the oxygen levels by adjusting dosing and adjusting reducing the aerators, because aerators, what aerators do is that because of the turbulence that they create, they somehow cause the oxygen nanobubbles that are released by diatoms to join together and leave the water body, right? Faster than ideally they should. So we want to have a very quiet environment. We want to have a quiet environment without too much turbulence, but we want some water movement, right? So that's the delicate balance which we are trying to strike.
Got it.
And
And lastly, when you reuse this water for, let's say, gardening purpose or flushing, I understand, but maybe for gardening purpose, could there be any negative impact to
in
the?
fact,
It could be a
I
positive.
think you will have a great for gardening. Let me go by application. For gardening purposes, when you have treated water which has low BOD, low COD, and high oxygen, there's a great benefit for gardening because the soil microbes will thrive on the oxygen in the water, right? So that's one benefit. The second benefit is that along with the treated water, there will be diatoms that are being injected into the gardens, right? Into the soil's upper layers. These diatoms will do photosynthesis on land and release oxygen into the pores, into the root zone, right, of the soil and the plants that are growing on that soil. So there is a benefit for the soil microbes in two ways. One, because the water itself has oxygen, higher levels of oxygen, and then you're continuing to produce more oxygen by photosynthesizing diatoms on the soil. So agriculture, gardening, lawn care, all of these applications will grow and benefit tremendously by the increased viability for soil microbes to function as a result of water from the STP. Now, if you want to use this water for applications like flushing and other domestic use, you may want to put some sort of a activated charcoal filter or a sand filter or a UV filter after the tertiary treatment as a final polishing step to make sure that it comes down the turbidity or all the different the aesthetics and some of the final polishing is done and the water is brought down to bathing quality, for example, if that is what you require for certain applications. So, it has to be done on a trial. You'll have to try it out and then put in those couple of additional systems in the end to ensure that optically the water looks as clean as you'd like, right?
Got it. Yeah. Thanks a lot.
Yeah. Yeah, hi, Kapil. Oh, sorry.
Yeah, Kapil was just speaking. There are lots more. This whole idea of health grade water, Arun, could you talk about that a little bit? Say if you have just black water, whatever, and it's treated through through diatoms to a certain level. What can we do then for us to achieve health grade water levels?
What grade water did you say?
Health grade water, drinking water, potable water,
or drinking water. I think is we are not really producing potable water, drinking water with our STPs. You will need to put in additional systems, UV disinfection systems, a particle sand filter, activated charcoal filter.
I understand that. So are you saying from your system, the water that's coming out of your system, just a simple WTP like a PCF ASF, PSF ACF, and a UV thing will take it to a health grade water level? Because that's not a very major cost in terms of.
No. I don't think it will take it to potable level. It'll be somewhere close, but I am not. Without some tests in that space, I won't be able to really give you a quantitative estimate.
Have you looked at things like this? Have you looked at taking it to a health grade water level, like a potable water level?
See, we have done it in the context of in lakes, for example. In lakes, sometimes you can be very close to bathing quality, right? So, or BOD of 3, right? In some lakes, we've been able to achieve that. COD.
Can you show us an example of what the size of the lake was, like how many million liters the lake was, and then therefore, what was it before you started your, your dosing, and then like what and how many, what was the time period for it to kind of get to a certain level, which is now bathing quality?
Yeah, I think can we discuss this offline? I'll have to dig up my studies on lakes and then get back to you on that.
I mean, just as a like a 2 minute thing, like if anything that as a for the audience also, like as a small little
Yeah.
example of
For
where
example,
you know.
there is a lake that is maybe a 2-acre lake that we just finished treating, that has let's say about 0.8 mld inflow of sewage on a daily basis, and it has about 17 days of retention time. So it has an inlet and an outlet. The outlet is bathing quality level, and with a fecal coliform level, around 1,000 or so. And this is even before, so this is roughly the range that I'm giving you. There have been other lakes that we have treated where the coliform populations are even much lower than that, right? When the full ecosystem is established. So I would need to just look at our data and slides and then get back to you on showing you those case studies. And I'll be happy to do that with anybody that is interested.
Sure. Thank you. Anybody else? I mean, of course, the conversation between you and Snehal and Srinivas ji is still pending, and that we'll keep for another session.
Yeah. We can, like I said, this since we are able to stimulate the growth of diatoms rapidly, this system can be extended to lakes, it can be extended to rivers, it can be extended to drains, nalas, practically any size, right? We can treat rivers, we can treat small systems. So, so we are, we are leveraging and completely restoring the aquatic ecology, and in the process, bringing all the various PCB mandated parameters down to the mandated limits. And depending on the existing sludge levels and the existing water quality, the time that it takes to achieve that mandated limits, could vary. It could be 2 months, it could be 4 months, it could be 6 months, it could be 1 month. So I think since we are working with nature and relying on nature to sort of heal itself, the amount of time that it takes to get there will be slightly different.
Understood. Bhaskar's also here. Bhaskar's also with a very has been championing diatom growth across very very many solutions. Bhaskar, do you have any last few? I mean, we are at 94 minutes now. Anything else that you want to add to this conversation?
No, nothing much. I think Sridhar has, Arun has covered most of the issues. We have to understand that diatom-based phyco remediation, we are merely substituting electric aerators with diatoms and diatoms. So, so electric aerators are consuming electricity, which is resulting in production of CO2, emission of CO2 at the power plants. Instead, when you use photosynthesis, you are consuming CO2 and nitrogen and phosphorus to produce the same oxygen. So that is a basic point. So if you lose track of that and get diverted into other issues, then it becomes very difficult to understand the system. The first step, please just understand this point. You are substituting oxygen from electric aerators with oxygen from photosynthesis, the two diatom algae inside the water. So this is more efficient than plants. Yeah, that's it. Thank you.
Thank you. Vinod Kumar ji has one last question, and we'll close after this. What's the pH level that's maintained in the system?
Around 7. Diatoms can grow across a wide range, between 5.5 or 5 to 8 nearly. So the pH would be closer to 7, right? So as long as the water, water that we are presented with has a pH that is more than 5, 5.5, it is a range in which diatoms can thrive. So We are quite happy with treating such waters.
Right. Okay. I mean, just as a closing thing, I would like to invite Niharika, who's worked with you, Arun, in a few projects, and see, basically, this is about the efficacy of the system in two spaces. One is the efficacy of the system as a standalone, as a treatment system. The second, obviously, is we are trying to also bring economic efficiency into these things. So as something that is I mean, as something that we're looking at, uh, like Bhaskar put it very, very eloquently, like this is a substitute for an electric aerator system, but it's not just that. I mean, uh, uh, while we are looking at digestion, sewage digestion at a whole, yes, anaerobic also plays a major part in this. Niharika, do you want to say something about this?
It was really a nice presentation, Arun sir. And all I can say, I think most of the parts has been covered. All I can say here is that if like those who wants to actually see this where it's working, so we have this in Bangalore only in two of our projects, which is one in Bumasandra and another one is Hostel Road, where we can have, I mean, we can enable the physical meeting and then you can go ahead and see the efficiency of how this remediation process is actually working. I mean, on the ground as well. So thank you so much, Arun sir, once again. And yeah, Nikki, that's something from my end.
Thank you, Niharika.
Thank you.
Thank you. And if there's anything anybody else wants to say as a last question or anything else?
Sorry, is this comes under phytoremediation, right?
No, it is FICO.
FICO, okay.
PHYCO. It's remediation. Phyto generally refers to plants, like in constructed wetlands or natural wetlands. But this is FICO.
Okay, thank you.
Because it relies on algae for treatment. So,
thank you.
Right. Anybody else would want to say something? Like, which now we're about 100 minutes in, long session. So close for the day. If anybody else.
yeah, thank you very much for the opportunity, and thank you very much for the discussion.
Thank you. Thank you, everybody. I'm going to shut the and hope we will kind of connect various people that who've been talking, including that Rajaramji and like others, to Arun. Yeah. Thank you. Bye bye.